who normally hosts these CERI seminar series. I'm Lady Liebenberg. I lead the immunology research program here at CERI. So yes, it's a new face for the online crowd. And I've asked Tulio to have me present our speakers, since I would like to fangirl just for a little bit. These seminar series, these are meant to include our superstar scientists from all around the world, bring them into a room to interact with junior researchers, early career researchers. I see Tulio entering the room. Thank you for letting me do the introduction today. Right. So this is no different from the others. We have two superstar scientists, leaders in the field of the genital microbiome here with us today. And I'd just like to take a little reminder of what these seminar series are meant to do, to expose us to a diverse research, expose us to research that might not be in our own field. A little bit of overlap, but it still gives you some new perspectives to help you solve your problems, maybe add to some, adding to the solutions required for another research problem, right? So this has been my research interest for a long time, understanding microbes, the immune system at the female genital tract, to help us try to design those interventions required to stop infections at the predominant site of sexual transmission in women, right? And then also to maybe treat infections that are currently there. And today it is my pleasure to host a few of the leaders within the field in this room. Two of them will be presenting in this forum today. So we have Dr. Jacques Ravel. He is the acting director for the Institute of Genome Sciences. He's a professor of microbiology and immunology at the University of Maryland School of Medicine in Baltimore, USA. We also have Dr. Caroline Mitchell, who's an associate professor of obstetrics and gynecology and reproductive biology at Mass Gen in the US. On the advert, but not here today, is Dr. Douglas Kwan, who is a physician scientist at Harvard Medical School. And he won't be presenting today. He's here in spirit. He does similar research to what we do, and Jacques and Caroline will be discussing it today. As I mentioned, these are leaders in the field of the genital microbiome. Look forward to hearing lots about the genital tract today and lots about bacteria and why it should matter to us to learn more about them. Welcome to Jacques and Caroline. Please share your wisdom.
Thank you so much, Lenine. That's a lot to live up to. So I'm a clinician, and we're going to start on the clinical side. Then we're going to talk quite a lot about the more genetic analysis, and then we're going to go back to the clinical side. So we're tag teaming just a little bit. So this is a picture of bacterial vaginosis. On the left is a gram stain. On the right is what we see through a bright field microscopy in clinic, although this is actually a phase contrast microscope, so it looks a little better. Bacterial vaginosis is this diverse community of bacteria. You can see there's lots of shapes, there's lots of colors. It looks very chaotic. And it is, in fact, chaotic for the vagina. This is not ideal. When you look at this from a sequencing standpoint, the top here, each row is a person, each color is a different taxa of bacteria. The top is people with BV, and you can see again, it's quite chaotic. There are lots of colors. On the bottom are people with what we think of as a more optimal vaginal microbiome, and it's just one color. So just one species of bacteria. And the species we think is ideal is Lactobacillus crispatus. You can see there's two colors though. So many people have Lactobacillus crispatus, but many people also have another lactobacillus called Lactobacillus inners, which is less optimal. So why do I care about BV? I care about BV as a clinician because it's quite bothersome to women, but we care about BV as a public health community because it's associated with many significant public health outcomes and a significant cost to the healthcare system. So it's associated with a doubling of risk for preterm birth, STI acquisition, including HIV, human papillomavirus persistence and progression to cervical dysplasia and cervical cancer. So big issues that impact women. And in Africa, this is a depiction of the vaginal microbiome in the fresh cohort in Umlazi outside of Durban. This is from Doug's work, and he's very sorry not to be here. And what you can see here is that the right half, the right side of this image, each column is a person, has that chaotic picture, lots of colors. It's a diverse community. And on the left-hand side, you can see a lot of green, which is that Lactobacillus inners, and very little of the gold, which is Lactobacillus crispatus. And again, we think Lactobacillus crispatus is the most ideal. However, you might be thinking to yourself, this looks actually a little different from the picture I showed you before, which was from US women. Here, there's a lot more inners. There's not very much crispatus. There's more diversity. So maybe is this just normal for Africa, right? Is the other picture just very biased towards the US? So I also ask myself that question. It's one of the things that comes up often in this field is what is normal and what is optimal? Are they the same? And this is a depiction of studies from around the world that associate a lactobacillus dominant microbiome. Many of these studies are old, so they're just using gram stain morphology of bacteria, but some are newer sequencing studies, so that's specific to Lactobacillus crispatus. But this lactobacillus dominance is associated with a lower risk for HIV, that's the red dots, lower risk for human papillomavirus is the brown dots, lower risk for preterm birth in purple, and a lower concentration of genital fluid IL-1 beta, which is a pro-inflammatory cytokine that hopefully you know a lot about because of Dr. Passmore's work. So what this shows, I think, is again, associations. Lactobacillus dominance is optimal for everybody, even if it is less common in some places in the world. So coming back to the clinical bacterial vaginosis, it's a very prevalent disorder, about 30% of people worldwide, and we are still treating it the same way that we did in the CDC guidelines in 1982. And that treatment is associated with a 60% chance of recurrence in the first 3 to 6 months. 40 years, 60% recurrence, no change in something that puts people at risk for cervical cancer and HIV. This is at a time when we are sequencing people's tumors to give them individualized chemotherapy. And we can't do more for women. We should be doing more for women. There is such capacity. And yet, what we have for women is the same treatment that we've given them for 40 years that doesn't work. And so I'm going to hand it over to Jacques to talk about how science is trying to move the needle on this.
I want to thank Lennon and Tolia for inviting us to talk. So what I'm going to do is kind of to describe. So Caroline did a great job at setting up the problem, and now we're using genomics to try to understand it better and come up with solution, which Caroline will talk about a little bit later. And so one of the things that we've done is try to build tools to basically crack that problem and better understand what's happening in from a functional perspective and compositional perspective in the vagina of the woman that we just that Caroline just presented. So back in I can't remember, like 2020, we published the first catalog of what we call comprehensive at the time, but we actually were wrong, and I wish we should put an erratum. It was actually not comprehensive. But it's a gene catalog of all the bacterial, fungal, and viral genes that are present in the vaginal microbiome. So this was kind of like going away a little bit from the traditional compositional survey, taxonomic survey description of microbiome. We were now moving into both composition and functions of what's happening in those microbiome. Okay, and so what this catalog really helped is to look at it from a functional view, but this time with resolution at the species level and sometimes even strain level. But the catalog was just not comprehensive enough, like I said, and so we came up with a new version of it, and you can see on this map, mass, you see that? You can see on the map, we tried to expand a little bit out of the North America for the source of the data, and we tried to expand a little bit, and now we have some data set from Africa. We had some, so we had some pretty strict criteria in terms of the depth of sequencing for those samples. So we actually worked with Margaret and Joni and some people at UNC in Zambia for as part of the Gates-funded project, the VMRC, to acquire those samples. So they were sequenced very deep, so which was very useful. We also have some sample from Bangladesh, and then we also gathered some of those samples that were collected and generated by other people around the world. So we set up a pretty extensive pipeline. I know a lot of people do bioinformatics here, so I'm sure they will understand all this. So now what we're doing here is shotgun sequencing. We're sequencing everything. One of the big problems when you do this on mucosal surfaces like the vagina is that 90% of your sequences, if sometimes more, actually could be human. Okay, so you have to sequence fairly deep to get enough bacterial data. So we have to remove human. We do a lot of quality filtering. We do de novo assembly of all the sample, and then we bin those samples so they can be actually taxonomically assigned. So what we do, we actually use at least 3 binners to do that. And then we put all this back together with MetaWrap to bring the best bin. So it optimizes all the bins for each of those, and then bring all this together. And then we eliminate any kind of potential contamination, and we generated about 15,000 mags, so metagenome-associated genome out of those data sets. Those mags generated 32 million gene. So total number of gene encoded in those mag was 32 million. Obviously, there's a lot of redundancy, as you can imagine. But we also used even the bin that were not actually all the reads that the sequences that weren't bin into those through the binner, because certainly low abundance in the sample, so we couldn't actually generate good bins. So that generated another 9 million read. And then we started, we pulled down a lot of genomes that were present in databases of microbes that are relevant to the vaginal microbiome. So that brought in another 4,000 complete genomes for a total of 47 million genes that actually went into building the catalog. And then we basically reduced the redundancy, and we ended up with only 1.1. It's actually a lot, because we actually doubled the size of the catalog, and now we have 1.7 million non-redundant gene. And so we started with over 2,500 metagenome, 15,000 mags, and now we have 1.7 non-redundant gene, and that's what makes Virgo 2. And Virgo 2 is actually it's actually a platform, so it doesn't come with just the catalog. So we actually deliver all the underlying data that was provided. So the metagenomes are there, the genomes are there, all the reads, the reads and the gene sequences are there, and we also have a lot of scripts that people can use it. So we want to make it a tool for discovery, but also a tool for analysis. So you can sequence your vaginal metagenome, obviously, or cervical vaginal metagenome, and you can actually process all that data through Virgo, and it gets you composition, function. And one of the big advantages of having something like this is that, a lot of time you do composition or you do function analysis from metagenome, but what people get is this big, what I call it, the functional soup, where you have, oh, this pathway is present in my metagenome, but all of a sudden you realize there's three genes in your pathway, and each gene comes from a different bacteria. This is completely artificial. It doesn't exist in nature. Those genes are inside each their own cells. They're not going to work together. So here, because we have taxonomy and function, we can actually do functional analysis by taxonomy. And that's actually what brings the value of this, because we can look at a bacteria and look at what they're doing. So the advantage, so we have a very good taxonomic annotation on all those genes. So now we raised it. We were about 60% in the previous catalog. Now we're at 91% of the gene in Virgo 2 that have taxonomic annotation, and almost 1.2 million are single species. So obviously, we all know genes can be found in many bacteria, so sometimes there's no way to actually say the gene belongs to one another. So then you have two copies. You have a copy that belongs to one taxonomy and a copy that belongs to another taxonomy. So, but we have all this information is encoded into and it's attached to each one of the genes that you find in the catalog. The other thing is that we have very good functional annotation. So we use a panoply of tool to annotate functionally each gene. And so when you sum it up all together, we have about 81% of the genes that are actually functionally annotated. But that also means there's 20% we just don't know what they're doing in the vagina. It's just kind of interesting. And so we were able to actually use this reference catalog. And to think about it is kind of a catalog of all the genetic diversity that exists in a vagina. So when we do the non-redundancy reduction, the redundancy reduction, we actually pretty strict. And so we allow gene variant to actually exist in this catalog. And this is why in a somewhat low diversity environment, you actually have 1.7 million genes. So the way to think about it, even though it's not the true definition of the pangenome, because pangenome usually is defined at the protein level, not at the gene level, here you can think about it of the vaginal metapangenome. So it's all the gene that you will find in the vagina. So, but you can still do those accumulation curve that people do for when they do pangenome analysis of specific bacteria. And you can see that for some species, we're getting close to saturation, meaning that every time we sequence a new metagenome, we're not really adding new gene to the pangenome of that species. But there are still species where we haven't reached the plateau. So there's still a lot of diversity to be discovered and integrated into the catalog. And one of them that I want to point to is Lactobacillus crispatus. And it's very interesting because we were kind of surprised. You know, we felt like we pretty understood very well the diversity. But one of the things to really point is that Lactobacillus crispatus is peppered with mobile genetic element. There is a lot of transposon, there's a lot of phage, lots of phage genes. And I think that's what's basically contributing to this constant discovery, because they are like a sink for genetic information. And so they're still in flux as a species. So the way you use Virgo is very simple. Now it's been implemented in a Python wrapper, and you basically take your read, your metagenomics or transcriptomics read, you just map them to the catalog, and we try to minimize the number of parameters that you want to play with. And then you use the script to compile that information into whatever you want, whether it's taxonomy, whether it's functions, and so on. Everything can come out of the compiler. And basically, at the end of the day, you get the gene in Virgo and the sample, and you get read counts that goes to each one of those genes. And then there's a whole set of tool that allows you to get to adding the functions, adding the taxonomy, and you'll see if you go to the catalog, there's about 11 files that you can actually combine to get the information that you're interested with. But the catalog also gave us an opportunity to start looking and exploring of like, what have we discovered? What's there into this catalog? So if you just look at it from estimating like composition community, this is about 3,000 metagenome that were mapped to the catalog. And this is the composition from a species level that you get from the catalog. And it replicates a little bit what Caroline has shown already. So remember, this is like a worldwide, and the geography is shown on top there. So you can see that there's samples come from all around the world, almost around the world. Unfortunately, South America is still not yet represented in the worldwide catalog. we remediated this, but it will go in version 3, I guess. But you got those that are dominated in red by Crispatus, in orange by Lactobacillus inners, and then you get this entire half of the graph that's basically this high diversity type of sample where you have a lot of anaerobes, and you can see the advantage of doing a catalog at the gene level. If you look carefully, you can actually speciate a lot of the different within the genus, like Gonorella, which has always said being Gonorella vaginalis. Now, when we do this kind of analysis, we can actually split it into 10 now described species for Gonorella vaginalis. So there's a lot of power in analyzing the metagenomic data. So the other thing that's some interesting information, like the de novo assembly and binning, it generated a lot of mags. And here, what I'm showing is the number of mag we got per sample. And you can see that in those communities where diversity is low, you get, a few mag, less than 10 more often. So those that are dominated by lactobacillus. But in those where diversity is much higher, you can see in a few sample, we got over 50 different very high quality bin. And those are only the high quality bin that we inputted in the catalog. Obviously, there's a lot more that's generated that's not as reliable, so they were not included in the catalog. So there's a very rich amount of information there. And this is the taxonomic distribution of all those bin. And you can see there's a huge bias. It represents what we see in all those samples towards Lactobacillus inners. And the other thing to mention is that once you dereplicate, Lactobacillus inners is actually also very diverse. And there's one genome and another, they don't look very much alike. Even if you look worldwide, it's almost like almost every woman who has inners has her own strain of inners. When Crispatus, it's not quite the case, but for inners, it is the case. And it's represented by this overabundance. But don't be fooled, Gonorella vaginalis is also very important, but now because we split it, it's split in 10 different species. So if you look at all those kind of like teal blue bar here, all those are Gonorella vaginalis. So if you sum it up, they get somewhat close to Lactobacillus inners, but not quite. Lactobacillus inners still is the most highly represented bacteria. So there's one interesting species that we found is this Lactobacillus portuensis. And this is an interesting species of Lactobacillus. It's found in Bangladesh, and it's only found in Bangladesh. It's found in a vagina of women who are actually dominated by Lactobacillus crispatus, and it's associated with those communities, but we still haven't found that particular Lactobacillus in the rest of the world. So biogeography exists and is important, and it's also why we really need to explore that biogeography. And when the whole symposium those past 2 days showed that there's such an underrepresentation of African sample where there's a huge amount of biodiversity that still needs to be explored. And the importance of this organism is still not understood. And there's a new interesting bacteria that I want to spend a little more time on. It's this Borrelia. It's a novel species that was previously undetected, and it's yet very highly prevalent. It's not highly abundant, but it's highly prevalent. And before I go on, I just want to show you a little bit a description of the diversity that we found. It's shown in this map, and it's nice because you can see all the discovery by major different phyla and different taxonomy, but also the size tells you how often do we find those bacteria, and we have a genome for those. So I'll talk about the Borrelia in a minute, but we discovered actually a lot of new species of known bacteria. So Phaniaceae used to be called Atopobium in the vagina. Now we have new species of Phaniaceae that are found in the vagina. And Prevatola is another one where we found actually species that are very prevalent around the world and were actually missed in because we only use the US catalog. And some of those species tend to be associated with also adverse outcome, those novel species. So important species to have included. But to go back to Borrelia, it's a very interesting bacteria. It's related to Agarthella, and I don't know if you read that literature, but there's been a lot of paper recently where And actually, one of the latest was by Ginny Marozzo for where they did an analysis very similar to what Doug did, looking at people, women in Kenya, I think it was, yeah, from the VOICE study, and they looked at acquisition of HIV based on what microbiome you had. And one of the organisms that they found, and so they did 16S, it was a Egerthella-like species, okay? And it's not Egerthella, it's Borrelia. And now we have its genome, and we have a lot of information on it. And so you can see its distribution. It's present in 80% of all those diverse community, which we call community state type 4, okay? But it's often present in very low abundance. But it's almost like a marker for CST4. Very interesting about this species is that it's asaccharidic, doesn't use sugar at all, okay? It relies on amino acids for energy production as well as formate production and CO2 and then pyruvate. So in the lab, they were able to kind of like recreate a little bit of the metabolism of Borrelia. And you can see that it can actually use acetate and lactate, which is very interesting, but only L-lactate, it looks like it. And it can actually generate putrescine, which is one of the compounds that's very odorific and associated with some of the symptoms that are looked for in bacterial vaginosis. And it can generate CO2. All this to generate energy for its metabolism. And if you look at some of the, we just picked like here, I think 5 samples where you can detect Borrelia and for which we had transcriptomics and that, is that species actually doing things in those samples. So you can see it's not very abundant, but almost every gene, like the genes are labeled here, the number here represent to all the genes that are in those pathways. Every single gene in every single one of those samples are actually they're present and they're active. There's transcript associated with those. So very, it must be a very active organism. And it's one of the things that I didn't mention, but we did in this catalog is one thing you can see is that when you look at transcriptome, there's bacteria that are often present in very low abundance, but they contribute a lot more transcript to the function of the community than some bacteria that are present in much higher abundance. So those low abundance bacteria actually could play a very important role. And we think Borrelia could be one of those. We also found some new diversity in Gardnerella. So there's 2 new species that we found. So one of them was only found in 3 samples, but the other one, it gives us confidence for its true existence because that particular genome was found in over 40 samples. So this is not just an error. It just was there. So the other thing that you can do with the catalog is start looking at tracking strains because you have this set of genes and you can start tracking the gene over time. And so one of the things that we answer with this single little experiment is this within person strain persistent, but people have done it with lactobacillus because you can cultivate it. You can actually do this very easily. And you can see that the same strain is often present over time, can switch, but often present. But it was not really known in non-lactobacillus dominated sample. How do those strains track over time? And you can see here in red is actually menstruation time. And you can see like during menstruation, those strains tend to disappear in abundance. But right after menstruation, the same strains of Gardnerella vaginalis, Gardnerella zvinzinski, even BVAB2, which actually Sharon described in a paper, Borrelia, Phaniaceae vaginae, all those are basically tracking over time and staying associated with the same host. There's very few actually changes over time. So those community, while they often appear to be less stable, the strains that make those community are actually stable over time. So the other thing that you can actually look at using Virgo is the virome or the virome. So here what we did is looking at bacteriophage. And one of the things that's a little challenging with bacteria and phage is that phage can exist inserted on a genome, but they can also exist as phage outside as, as phage particles. Okay, so here what you can see is we plotted the relative abundance of bacteriophage. So total genes are associated in to bacteriophage. And you can see that it's very interesting. Actually, Crispatus, like I mentioned earlier, is peppered with a mobile genetic element, including phages. And it's actually the communities where we see actually the lot more phages than in the other communities. But one thing that was interesting is that we tried to look for those that were basically virulent, so phage that are lytic versus phage that are lysogenic, that integrate and just stay in a genome. And when you plot, so from the sequences, you can actually tell if a phage is virulent or temperate. And so when you plot the ratio of virulent to temperate, what you can see is all those samples, and unfortunately, it shows that it might not be a super prevalent mechanism or occurrence of lytic phage. Only 63 out of the 2,000 something sample actually had a higher ratio of virulent phages compared to temperate phage. Okay, so that's only 2.5% of all the sample, and the majority of this community were to CST4. So even though Crispatus has a lot of phage, they don't appear to be lytic phage. But those lytic phage appears mostly in community of state type 4. Okay, and so there was a very when you see this ratio high, you can think of it as a lytic phage just starting to bloom and started to infect and generate more phages. Okay, so we call this like potential bloom event. And so we started to investigate this. And as part of this data set, what we found is that there was 5 samples that actually came from the same woman. They were collected over time, okay? And so you can see those 5 samples, they were all there, and I plotted them, and you can see that at time point 4, for example, that's where you see the most phage, probably lytic phage, that are present there. So, and if you look at the longitudinal pattern of this woman over time, you can see that in green is Lactobacillus gasseri, okay? And so we were thinking like maybe what we have is a phage that could infect Lactobacillus gasseri and is responsible for its kind of like cyclic and loss of abundance over time. So it's killing Lactobacillus gasseri and then it's blooming as a result. Okay, so we tried to look for this, and we did a lot of experiments, tried to filter the sample. Remember, those samples were frozen. We filtered the sample. We tried to infect gasseri because we have that strain growing. Just could not get any infection. Okay, so my PhD student decided to go and look by electron microscopy of all those filtrate, and he found phage. But the problem is, like, he called it a graveyard of fragmented and mangled phage-like particle. So basically, the frozen process, the freezing process was a big problem, and all those phage are basically dead. They're just sitting there, but they're there. So we have actually evidence that phage can happen, that lytic activity actually happen in the vagina. And I think this is one of the first evidence, even though we don't have the biological evidence, we have at least a microscopic evidence. And now that we've actually seen the phage, we can actually go and try to find them in more in fresh sample. So that's going to be something that I think a lot of people are going to be interested in doing. So the other thing that I just want to finish is that you can use it for transcriptomics analysis. And I think transcriptome is really the future if we really want to understand what's going on in a vagina. And the reason I'm saying this is that on top, so here what you have two plots, one that represents metagenomic data, and it's basically compositional data. And at the bottom, what we plotted is the relative contribution of the transcriptome of each bacteria to the transcriptome. So it's how many genes they express and that those genes contribute to the transcriptome. And what you can see is that there is some correlation, but there's not that much, especially when you start going into those communities that have a lot of Gardnerella. So it seems like you have a lot of Gardnerella, but that Gardnerella is not actually doing that much. A lot of activities is coming from the elder bacteria. So, and which means that just because you have a lot of Gardnerella doesn't mean Gardnerella is actually important. Maybe other bacteria might be actually more important than Gardnerella. And this composition is basically leading you maybe in looking at things that might not be as important as those that are actually doing things. So, but you can actually start looking at this. And what Michael France and my lab did here, he took those all those 300 or so transcriptome that we had, and he mapped them to Virgo, and he clustered them with this PAC map, which is a little bit an improvement on UMAP, but it's just generate this embedding. And he basically, you have two clusters, and it's one cluster, you have all those community state type 4, plus some of the community state type 2 that are dominated by GESRI. And the other one, you have all the other community state type 1 dominated by Crispatus, 3 by Inners, and 5 by another lactobacillus called Lactobacillus jensenii. Okay? And then what he asked a simple question is like, can we look at the expression of all those genes that basically deal with carbohydrate utilization, so the KZ enzyme. And he plotted them like this. And so on the left-hand side, you can see the fold change of all those genes, okay? So the expression of those genes in cluster 1, so in those community type 4 type things, and in cluster 2 on the right-hand side of that graph. And they color-coded by what they're doing. So some of them will degrade glycogen, some of them will degrade mucin, so the glycan that are on mucin, or like sialidase, for example, is present there. And what you can see right away is that community type 4s are a lot better equipped at doing all those functions than community type 1 and 3s are, okay? And one thing that I want to point to is this GH33. That is a sialidase. And sialidase are very important because they can cleave off that cap, that sialic acid cap on the glycan that are attached to mucin, and then make those sugar, those glycan available for degradation. And sialidase is mostly actually expressed from a species of Prevotula. Like over 50% of all the sialidase are actually coming from Prevotula. Gardnerella only contributes maybe like 10% or so of all the sialidase. And you can see that sialidase are completely absent from the cluster 2. So you can start really gaining a lot of functional information by using transcriptome, and using Virgo, you can actually have visual, basically representation of what's happening in the vagina. So I just want to finish with one more thing. The title of the first paper was, we talked about within women intra-species diversity. And so what we did in that, what we discovered in analyzing the first catalog is that when you look at communities that are dominated by one species, okay, usually the dogma, and I've always thought that way. I was like, well, there's only one strain. One species, you see like it's dominant, and it's just like a culture. You have one strain, okay? Well, it's actually not the case. What we found is that there are actually multiple strains of that species that coexist within those communities. And the way we found this is that if you take a metagenomic sample, okay, and you map this to the catalog, okay, so you have all those genetic variant and functional variant, okay, and what you get is about 3,000 genes, 3,300 or so, that's going to belong to Lactobacillus crispatus, okay? But the genome of Lactobacillus crispatus of one strain only encode, in average, about 2,000 genes. That means there are genes that are present that just cannot be encoded on one strain. That means you have another strain that brings new genes that add to that 2,000, and another strain that start adding more and more genes. And then you have this basically set of what I call a conspecific genotype. They're the same species, but they're different genotype, different strain. So you can say strain of the same species that exist in the vaginal microbiome. So this was a very important finding, and which we actually leveraged to do what Caroline is going to talk about next. And the last thing, I've presented this figure on Tuesday, is the fact that while there's diversity within a sample, there's also diversity across the globe, and it varies by species. And here is just Lactobacillus crispatus. And what you can see, the color coding is by kind of continent here. We put it that way. And what you can see, we find species that are found everywhere in the globe of Lactobacillus crispatus. So you can find I said species. lineages type that are found in Africa, North America, Europe, and so on. But there are also lineages of Lactobacillus crispatus that are only found in certain geography. And I think this is also a very important point to consider when you design potentially like a live biotherapeutic like Caroline is going to talk to us about. So this is where you can find, well, actually, this is not where you can find, I put it on the previous slide. And the version 2 should be available in about 2 weeks on the site. And this is all the file, the thing I told you about, that you can actually annotate all your genes and so on. So there's a very nice tutorial, and you can actually replicate this pretty nicely. And with that, I'll pass it on to Caroline.
So now that you've learned all about what comprises the vaginal microbiome, the different species, their different function, the question you might have, the question I have for myself is why? Why do some people have crispatus? Why do some people have that diverse community? What is it that determines that? In the gut microbiome, we know it's diet. We know different times of day, different things that impact the gut microbiome. How do we know that? Mice, right? You can take a mouse, you can feed it different things, you can see how that changes the microbiome. There is no animal model of the human vaginal microbiome. We are the only species that have lactobacillus dominance, which is scientifically very interesting and also scientifically very frustrating because we can't do the like very specific knockout a gene in the mouse and see how that impacts the microbiome. We can't do that. What we also see after the antibiotic treatment that I told you does not work, we don't see that people colonize with Lactobacillus crispatus. They have Lactobacillus inners, which again, Jacques told you, has sort of the most genetic diversity. Everyone has their own Lactobacillus inners, but why that one? Why is that the one? The way to find out causality is to do interventional studies, right? You have to do an intervention, you have to pull a lever and see what happens. We have to do that in humans, unfortunately. So we're using all of the knowledge that Jacques has generated with Virgo and Virgo 2 to help us figure out what type of intervention will allow us to sort of shift the microbiome. And our goal is to shift it towards Lactobacillus crispatus, which seems to be the optimal no matter where you are. So there does exist a single strain of Lactobacillus live biotherapeutic product delivered vaginally. And in a phase 2 trial, when you deliver this single strain product vaginally after antibiotic treatment, a significant proportion of people colonize with Lactobacillus crispatus while they're using it. After they stop using the live biotherapeutic product, about half of people lose colonization. So that single strain is probably not ideal for those people. And I think Jacques' work show you that like there are a lot of strains of crispatus. Often people have more than one strain. So a single strain product may not be the right answer. So what about a multi-strain Lactobacillus crispatus product? And we're going to talk about that. That is a study that's ongoing here in South Africa with Disebo as one of the PIs funded by the Gates Foundation. I'll say the multi-strain product assumes certainty. It says, Jacques' genomic work, we know exactly what we need. It's all crispatus, it's multiple strains. I'm going to show you how we pick them. We are certain. And he showed you that the functional bacteria may not be the ones that we see, right? The ones we see the most of may not be the most important. And so the other approach one could take to try and shift the microbiome is to be to say, let's just put it all in. And so that's vaginal microbiome transplant. It's taking the community, whatever is there, whoever is there. We're not putting a bet on any, well, we are going to put a bet on crispatus, but along with crispatus, we're not going to put a bet and we're just going to put it all in. And these two approaches, I think, are going to help tell us more about the why. Is it the community? Is it the host? Do we have to match community and host in some way? The Vibrant study is 2 live biotherapeutic products that are multi-strain Lactobacillus crispatus generated using an algorithm and Jacques' data. these specific, and I'll show you that. So based on this idea that in a Lactobacillus crispatus dominant community, there is more than one strain. And so in order to get enough sort of coverage of the metagenome, you need probably at least 6 strains to get sufficient coverage of that whole metagenome. So from collaborators in the Vaginal Microbiome Research Consortium, some of whom are in this room, we identified isolates of Lactobacillus crispatus from people with a stable Lactobacillus crispatus dominant community. So theoretically, the best Lactobacillus crispatus out there. 15 genetically unique strains were obtained. And then a computational algorithm was used to select the combination of 6 that best replicated the metagenome in the most number of people with the stable Lactobacillus crispatus community, which is a lot of qualifications. And because that is only covering the metagenome in some of those people, we also said, let's just put all the 15 strains together and see if we need more redundancy to help more people. And so this consortium, there were multiple 6-strain consortiums that came up, and then Jacques in his lab tested those consortia for antimicrobial activity against BV-associated microbes, and we picked the one that had the most activity against BV-associated microbes from the US and from South Africa. So these are now being studied in a phase 1 randomized controlled trial being conducted in Boston in the US and also at the Vulindlela site for Caprisa, just outside of Durban. DeSebo and her team have been doing an excellent job of enrolling participants. We are a little bit farther behind, but on our way. And then Lenine will be doing a lot of the immunologic analyses for this trial. Caprisa serves as the data coordinating center for the trial. And Sinai, who's over there in the back of the room, will be doing the 16S sequencing analysis for this trial. So it's a very Africa-based study. We're comparing multiple dosing strategies of the 6-strain consortium. We thought the 15-strain consortium would take longer to produce, so we only put in one arm, but the company that produced it for us did a great job, and they were both available at the same time. Our outcome is colonization. It's detection of one of our strains at greater than 5% relative abundance in metagenomic sequencing, which is a low bar. If you've seen Lactobacillus crispatus in the figures that we've shown so far, when it's there, it's usually there in very high abundance. And so hopefully that's what we're going to see, but we set our bar low. So this study is going to hopefully answer a lot of the whys. Will the same isolate colonize in all recipients? Is it the bacteria? Is there a super Lactobacillus crispatus? Or does it matter who the host is? Will the South African Lactobacillus isolates in the products colonize better in the South African recipients and the US ones in the US folks, or does it not going to matter? And does our response, we are treating people with antibiotics ahead of time, ideally to sort of knock down the quantity of the BV-associated microbes. Does the response to that antibiotic treatment, does that matter? Do you have to clear the field, or can the Lactobacillus crispatus clear the field itself? This is a small study. It's a phase 1 study. We may not get definitive answers to all of these questions, but this is the interventional study that starts to uncover the biology that we do not yet understand. We're describing it very well. Jacques is describing it very well, but the causal links, what is actually happening, we still don't understand well enough. That's the approach of certainty. This is the approach of uncertainty. This says we have no idea. So it's the first randomized trial of VMT for recurrent bacterial vaginosis. So this is in the people who have that recurrence. The antibiotic therapy they've used over and over and over, and nothing is working. We're taking vaginal fluid from healthy people and giving it to other people. So obviously the number one concern is safety. And so we screen our donors quite thoroughly. We screen each individual donation for human papillomavirus. And also we have put a bet on crispatus dominance. So we do want the donors to be crispatus dominant. We want each donation to be crispatus dominant. People donate fluid by using a disposable menstrual cup. And we test donors before, at the end, And then we test again a month after they finish donations to make sure that we haven't missed an acute infection late in the donation period. So we hope that this is making it as safe as possible. The recipients, again, are people with a history of recurrent BV, so nothing has worked for them. They receive antibiotic treatment and then 2 doses of transplant. And I often get asked, why 2? What made you pick 2? This is the first study, so it was a guess. We also, if it works, I want to know what worked. And if I put more, 5, 10 doses in, then I'm not going to know which one has the special sauce. So that's why we chose 2. And we deliver it it's very basic, just a transfer pipette. It's not fancy. So these are our results from a pilot study. So the FDA required us to do a small pilot study to demonstrate that we could do this safely. So it's 8 people randomized 1-to-1. The top row there is our placebo recipients. The bottom row is our transplant recipients. Green is Lactobacillus inners. Gold is Lactobacillus crispatus. Blue is Gardnerella vaginalis. The red triangle is when people got antibiotics, and the 2 arrows are when people received transplant. And we followed out to 6 months. So you're seeing every column is a time point. And what you can see on the bottom is that at 6 months, our transplant recipients, 2 out of 4 had crispatus dominant vaginal communities. And these are people in whom nothing had worked before. I'll say 1 of the other 4 symptomatically is totally better. And then the 4th one, she definitely is not better. On the top, none of our placebo recipients achieve crispatus dominance. So this to us, 50%, you might be like, yeah, 50% is really not that great, but 50% in a community that is usually 0%, we think is pretty good and is very promising. And when we look, so from everybody who had detectable crispatus, we cultivated isolates, we sequenced those isolates, and then we looked at the genomes on a tree. So the blue here is our reference genomes. The gray is genomes of crispatus from the placebo recipients. Purple is the VMT recipients, and orange is the donor. And I think you can see the orange and the purple or pink, whichever color it is to you, they're totally undifferentiatable. They're the same. So it really is transfer of a crispatus isolate, at least 1, maybe more. Our metagenomic results are coming. So it is transfer. It is, in fact, a transplant and an engraftment. So this study also is going to answer, is it the community? If I transfer a good community from one person to another, is it the good community? That's all they needed. Or do you have to match host characteristics with a community? Am I going to have to match donors with recipients like you would for a kidney transplant? Right now, we're trying to be very agnostic. We're not matching anything. This is the question, is do you need to do that? And this is going to help us ask the why. Why do some people have inners? Why do some people have crispatus? Is it because they just weren't exposed to the right lactobacillus? Or is it that you have to have a match? Or is it something about the host that they just are never going to colonize with lactobacillus? And this, again, is the mechanistic biology that we have not understood thus far and that interventional studies are necessary to start to uncover. So in clinic, I feel like I'm the Wright brothers. People come to me and I say, I have the same antibiotics I had in 1982. That's what I have for you. And I know they don't work, but let's try them. And what women deserve at least is Star Trek. Probably more than Star Trek, but at least Star Trek. And that is possible. So yes, thank you for that. That is what we need to do. Jacques and I both work with really amazing teams and could not do it without all of those folks, many of whom are in this room. And so thank you for your attention. And I think we're happy to take any questions.
Thank you so much, Caroline and Jacques. What did I say about leaders, hey?
Yeah.
We started off with such a bleak picture of, oh my God, what's going to happen to our women? We're never going to fix this. And throughout this presentation, I just got more excited and excited. We're learning there's some new knowledge that's going to be incorporated into a solution that's going to fix us all. Thank you for your contributions to this. Not only you, there are many people within this room who have contributed to this work. It is amazing to see what has been done. And that there's a path for more to be done here, right? I guess the floor is open for questions. Oh, I've got hands. There's a roving mic, I think. Oh, there we go. Use this mic over here.
So I've got excellent talk, guys. Two questions, one for each of you. So the transplants you do are they timed in relation to the menstruation cycles, or you do it?
That was the original idea. It turns out it's very challenging in menstruating people for some people to accurately predict when their menses are, because we were like, we'll do the baseline visit right before the period. They'll take the antibiotics during their period. Then they have the transplant, and there's 3 weeks, 4 weeks until the next menses, and so like the transplant can establish. That is aspirational. Practically, that has been a little more challenging. So we try and do the transplant sort of soon after a menstrual period so that there's plenty of time until the next one, but it's been a little more variable than I initially planned.
Great, thanks. And then for Jacques, the metatranscriptomics, do you only use Virgo 2 as the database to compare against, or?
Yeah. So for the transcriptomics, when we process the sample, when we're interested in bacteria and phage and fungi or even trichomonas, we use Virgo, but big advantage. So it's a disadvantage in metagenomics to have, well, I don't know, you waste a lot of sequencing on human, but you can still use that human. So don't forget to consent if you ever want to do studies like this, consent for using this, because even with a metagenome deep sequence, you have enough information to capture a lot of genetic variation there. And you can also look at the host transcriptome and in transcriptome, because now there you get about 50-50. Even though it's a little more challenging because the host in community type that are very diverse tend to be a little lower, though you get less coverage of the host. So, but you can look at the host as well in those samples, but we only use Virgo. Yeah.
So, hi, I'll give it to you just now, Joanne. I have a question that involves both Jacques and Caroline. For the lady who had the VMT. Okay. 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 And the lady who had an intractable, BV state, would you be able, from Jacques's, interpretation of the low abundance, but really amazingly dysfunctional bacteria that contribute to Barriella or whatever, would you be able to go back and look at that lady and then make a call about these women that have this particular low abundance and being irretractable for VMT, maybe? I don't know. Are you planning to do that?
That's the idea. I mean, I think both of these interventional studies will allow so many of those questions to be asked. Like, if it doesn't work, if either type of intervention doesn't work in someone, is it person characteristics? Like, can we compare between people? Oh, the people who are blood type B doesn't work in those because our donor was blood type A. Or is it that everyone who it doesn't work in has this particular microbe? That's what it might be. It's sort of this, I think there are so many questions that we all want to ask about what determines success, what determines failure, what determines sort of the persistence of a microbial community. All of those you can only ask, I think, in a interventional study.
Can I have two questions? Because I have to choose if I can't. So the first question, Caroline, I always absolutely love what you do, but both my questions are for Jacques. So the one was with Virgo 2, and it might be in the manuscript and I've just forgotten, but can you comment when you added data, when you kind of between Virgo 1 and Virgo 2, and you added data from different countries and different regions, like how much did you gain by adding each new additional site. And the only reason I'm asking that is, you know, like with all the genetic diversity within Africa, Zambia can't represent the whole continent, but by the same token, I'm sure South America would say the same. So, you know, it was just like about how much extra are we gaining by adding each additional site from your experience?
Yeah, so actually we didn't look at by site, but I mean, just by adding the sample, and it's still dominant. There's more sample coming from the US. We doubled the gene catalog, the size. So I anticipate that there's still at least another doubling to happen by adding more sample, like from South America, a lot more sample from Africa. I'm sure
could that be an analysis your lab does on the data you already have?
Actually, you know, we never looked at it that way, but I think after the symposium and everything, it's something that we will add to the paper, like the contribution and to the diversity. I think that's a great idea.
And then my second question, which could straddle both Caroline and Jacques, but I noticed one of the plots you showed where you had really intensive sampling over menstrual cycles, that G. vaginalis dropped during menstruation, abundance of G. vaginalis. And that's so counterintuitive because everyone associates menses with diversity. But what you showed very clearly there is that G. vaginalis dropped. And I was wondering about studies that you knew about or had done that looked at, you know, the total biomass change during menstruation, and maybe, you know, with the flushing out of menstrual blood, you lose a lot of the biomass, and that's why lactobacilli don't survive, but Gardnerella is clearly not surviving either. I was like amazed by that observation.
Quantitative analysis during menses is really challenging because it's already challenging outside of menses, because unlike stool where you have like 1 gram of stool, there's X amount of bacteria. In vaginal sample, it's either like some amount of liquid, potentially like fluid, if you can actually measure it accurately, or it's a swab. So you refer everything to a swab, but what does a swab represent? And you know, in comparison to the vagina, we don't really know. So in menses with flow, with all the challenges, I think it's really hard to be extremely quantitative. But one thing that's interesting, and we've done the description of the daily changes over time, and this is just one of them, and it was not daily, but the Gonorrhea and everything, what you see is when a woman is dominated by lactobacillus, they lose that dominance during menses. But if they don't have lactobacillus outside of menses, you can see major changes during menses. Like there's still, and often there's a few bacteria that tend to dominate, and you can see the big shift. Like, and Gonorrhea is actually not, a species that's dominant during menses. I mean, you can look. We have like 165 or so of those, and that you don't see Gonorrhea just blooming, even in crispatus. Yeah, makes sense. Yeah. Yeah, but it's still an issue. Like, what does you collect a soft cup? What does that represent compared to what's in the vagina? We still don't know that. There's still challenges.
We're almost out of time.
Okay. My question goes to Jacques. My question is on host DNA depletion. Yeah, I've seen in your presentation the depletion was only done after sequencing. So my question is, were the samples treated before extraction? Okay.
No, so it's an eternal challenge that a lot of people are trying to resolve, and I don't know of a solution for this. If you were to do like filtering for human cells versus, but Caroline showed you I would say beautiful, but those what we call clue cell, which are human cell that have tons of bacteria aggregated to it. If you start filtering human cells, you're removing those bacteria at the same time. Spinning will do exactly the same thing. You spin down like low spin to get the human cells, those bacteria will come in as well. So differential lysis people have tried, but there's no bacteria that won't lyse when you lyse human cells. So you're already like removing bacteria that you want to know. So every methods that have been tested so far introduce some level of bias. And then what you're doing, you're modifying your bacterial communities, and then you're not looking at really what's at the real picture. So for now, the approach is just sequence your way through and embrace that sequence and make use of it, and then it's a win-win on both sides. So for vaginal metagenome, we usually do between 40 and 50 million paired, so 100 million total. So it's, yeah, for one sample. Yes. So no, you're getting the X plus. With the X plus now, it would be even cheaper for the same depth, if not more. So it's, I think that we're getting to a point where it's the cost of trying and even the cost of the method that you would use to do this is going to be more than just the cost of added sequencing, because sequencing cost is just dropping constantly.
Okay, so I'm just going to squeeze one quick question. Given the substantial variation in microbiota over time and space, what are the chances that a probiotic may be applicable to people with different genetics, perhaps consuming different diets? Will this be a one-shot? Do we need to change the approach to various situations?
That's why we're doing the study. Right? Like
They're starting off slowly.
Yeah, we can hypothesize, but without a causal model, the reason people know more about this in the gut is because they feed mice different bacteria and they see how long they stick around. And without mice, you have to try it in people. So I think, again, we have so many questions like that these first studies, it is really just a best guess, right? Like the way we did things, why 7 days of dosing for our live biotherapeutic? We give antibiotics for 7 days. So it seemed like a good number, right? And it was like not too long, not too short, but it's all the best guess until we start to get some actual data.
There you go, Tulani, that's a start. And would there be any effect of the transplant along with contraceptive use?
Also a great question. With 8 people, only 4 of whom received transplant, I can't answer that currently. We do know that in epidemiologic data that hormonal contraceptives are protective against BV, but that's cross-sectional epidemiologic data, and so I don't know.
Okay, that's a start. Thank you very much to our presenters today. Thank you everybody in the room and online. We have a closing remark by Tulio before we head off. Thank you again for being here.
No, yeah. Yeah. My closing remark, it's very simple, and thank you so much. I thought that today was absolutely fascinating, like how you guys went from like, what's the problem? What's the clinical problem? And you got so in details of the bioinformatics and the genomics, and then brought back to the problem and almost getting a solution. Yeah. I think that was really inspiring today to see how groups can work together from the clinic to the very advanced bioinformatics to solve a big problem. And we felt really, really fortunate to have you two here. Yeah, and it's fantastic. I think that the level of the seminar is getting better and better. Yeah, it's just phenomenal. And thank you everyone for coming. It's great to see a full room. Yeah, that's why we're bringing all these seminars here, is to really expose you what is really world-class science. Thank you.
Very much. Now, some snacks outside. All right, and for anybody who needs the CPD points, you just need to sign register outside.